Good morning, students. We will continue the topic of protein metabolism, computation number 5.4. Today we will see the remaining part of catabolism of amino acid. In the previous competency, we have studied how ammonia group is removed from the amino acid. Now, after removal of amino group from amino acid, the carbon skeleton is remain, and that carbon skeleton may be in the form of pyruvate, it may be in the form of intermediates of TCA cycle, or it may be acetyl CoA. Now, amino acids like alanine, serine and cysteine, on removal of amino group, that is deamination, they form pyruvate. Now, this pyruvate then converted into oxaloacetate by carboxylation reaction, and this oxaloacetate may be converted into glucose. Or this pyruvate undergoes oxidative decarboxylation to form acetyl CoA. This acetyl CoA either enter into the TCA cycle, or in fasting state these acetyl CoAs are utilized for synthesis of ketone bodies. This pyruvate also undergoes transamination reaction to form alanine, so depending upon the requirement of the cell. So these are the various fates of pyruvate. So pyruvate is formed from three amino acids like alanine, serine and cysteine. Now coming to the other amino acids. So some amino acids, as I have told you, forms intermediates of TCA cycle. Like if you see on the right side, proline, arginine and histidine, they get on transamination forms glutamic acid. Now this glutamic acid on deamination forms alpha ketoglutarate, and this alpha ketoglutarate joins the TCA cycle at this stage. Similarly, some amino acids like valine, isoleucine and methionine forms propionyl CoA, and this propionyl CoA forms succinyl CoA later on, and this succinyl CoA joins the TCA cycle. Other amino acids like phenylalanine and tyrosine forms fumarate on deamination and joins the TCA cycle. Aspartic acid on transamination form oxaloacetate, or on deamination form oxaloacetate, and this oxaloacetate joins the TCA cycle. Other glucogenic amino acids, they also forms oxalate, oxaloacetate and joins the TCA cycle. Similarly, the ketogenic amino acids, mainly leucine, forms acetyl CoA and enter into the TCA cycle in this manner. So in this way, the remaining carbon skeleton of amino acid is utilized. So as I say that leucine converted into acetyl CoA, and this acetyl CoA is then converted into ketone bodies or TCA cycle. Some amino acids forms two ketoacids like isoleucine, phenylalanine and tyrosine. They form acetyl CoA as well as intermediates of TCA cycle. Tryptophan also forms two ketoacids. First, tryptophan forms alanine, which forms a ketoacid pyruvate, and tryptophan also form acetyl CoA. Now up till now we have studied deamination reaction. Now we will study decarboxylation reactions. Decarboxylation reaction means when carboxyl group is removed from amino acid, they forms amines. Normal amino acids forms monoamines, whereas basic amino acids which have two or more amino group, they forms diamines or polyamines. So these are the examples of biogenic amines. Like serine on decarboxylation forms ethanolamine, which later on forms choline. Tyrosine on decarboxylation forms monoamine called tyramine. Dopa on decarboxylation forms dopamine. Dopa is basically formed from tyrosine. Tryptophan on decarboxylation forms tryptamine. 5-hydroxytryptophan, which is the derivative of tryptophan, on decarboxylation forms serotonin. Histidine on deamination forms histamine. Ornithine is a basic amino acid, so it contains more than one amino group. So on decarboxylation, ornithine forms polyamines, and the examples of these polyamines are putrescine, spermidine and spermine. So these have very important role. Remember the names of polyamines whenever you are asked in Viva. So how they are formed? They are formed from decarboxylation of basic amino acid ornithine, and the examples are putrescine, spermidine and spermine. Lysine have two amino group, therefore on deamination it forms diamine cadaverine. Cysteine on deamination forms taurine. So this is about the fate of carbon skeleton and decarboxylation reactions.